clinicians, scientists, and the lay public all have an appreciation of the fact that heart disease is prevalent and devastating. And this is partly because the adult mammalian heart, including our patients' hearts, have very meager capacity to regenerate themselves by laying down new cardiac muscle cells to replace the ones that have died. And although there has been some intriguing progress in showing measurable amounts of restorative growth, that's still quite meager, even by all the identified mechanisms working in concert, and clearly not sufficient to restore the loss of cardiac muscle cells that occurs in heart disease. And so the question then becomes, what might be fruitful routes to solve the clinical problem of heart attacks and chronic heart failure viewed from the perspective of being a muscle cell deficiency disease. The work of my lab here in London continues to focus on dormant stem cells of adult myocardium. Conceptually, these are not just another cell therapy product lined up along with half a dozen or more competing cell therapy products. These are potential targets for activation of self-repair in situ. And so the grail is not just having better, smarter cells to graft. The grail is to find the cell that's a target for activating signals to promote self-repair. And that's the context in which the experiments are done. What sets all of this aside from the cells with agreed potential for cardiac muscle creation, like ESLs and iPS cells, is that these guys are in the heart already. They're in the right place. Molecularly, they are part way towards converting into cardiac muscle, though they don't do so spontaneously. Unlike cells from the circulation or bone marrow, these guys already expressed many of the heart-forming transcription factors that are essential to create a cardiac muscle cell in the early embryo or in differentiating embryonic stem cells. We were able to show that if injected into the bloodstream of a mouse with an experimental heart attack, the cells would home to the area of injury. They would seamlessly interdigitate with the injured muscle, and they would become cardiac muscle with some measurable frequency, making a significant contribution when delivered that way to the number of cardiomyocytes in that border zone between the infarction and the host myocardium. And for us, this was an interesting finding, but it raised as many questions as it answered. So we began our effort to test multipotentiality by trying to pinpoint the cells that were clonogenic and then ask if they were cardiogenic, and if they were cardiogenic, could they do other things too? And that work during my first years here at Imperial took us in the direction of an ancillary marker referred to as the side population, or SP, di-efflux phenotype. And what we found is that the side population strongly predicts the cells within the SCA1 population that are clonogenic. And what we then went on to do was not just make clones and score the frequency of making clones, but actually to grow up the clones, characterize them rigorously through a variety of molecular techniques, usually profiling 45 different genes in each of 24 clones at each of multiple stages of their maturation. And from our analysis of the injected clones, we were able to show that these cells from 4 representative clones all could form cardiac muscle, smooth muscle, and endothelium, that injecting the clones had marked effects on the ability of the infarcted mouse heart to contract. Its ejection fraction was improved. Infarct size, although the same at one day, was reduced relative to the healthy myocardium 12 weeks out. And all of this was true even though the number of surviving donor cells that persist to 12 weeks is vanishingly small. So one of the paradoxes in the field of cell therapy for heart repair is that in most of the animal models, and likely in patients as well, with the trials that are now ongoing, the durability of cell grafting is evanescent. It's fleeting. The cells are there. They make things that make the heart better. But even though the surviving cells turn into heart muscle and blood, the number of surviving cells under current conditions of delivery is very, very few. So what they make may become more important than what they turn into. And this can take us down the line in a number of different directions of relevance to patients. One of them being find out what they make and give that. Because those hormone-like proteins or other kinds of signals might have direct relevance to promoting heart repair. And alternatively, in collaboration with material scientists and bioengineers, a tissue engineering strategy to promote cell grafting with encapsulated cells, sheets of cells, or three-dimensional constructs. All areas of activity in the AHA meetings might have a strong likelihood of promoting more durable engraftment, more maturation of the grafted cells, but it will require something different than the current trials of injecting a naked suspension of cells and relying on them to carry the full weight.